My name is Shawanun Pratgao.
And my name is พิชยาภาแก้วสมบัติ
We are the students from Princess Julapon Science High School, and we are your moderators today. First of all, we would like to introduce our commentators in this room. The first commentator is Assistant Professor Dr. Wuttipong Mahakam from Khon Kaen University. And we also have Dr. Shontisha Jinapon from Le Rashapat University as our second commentator. Today we have 17 schools and 17 projects that are joining us. And now, we would like to introduce 10 schools that will be present in this morning sessions. When you hear your school name, please raise your hand. The first school is Princess Julapon Science High School. Okay. Second, Princess Julapon Science High School Trang. Third, Furukawa Raymond Junior and Senior High School. Fourth, Pathum Vilai School. 5. ผาแดงวิทยาลัย School. Oh. 6. Demonstration School of Khon Kaen University Secondary ศึกษาศาสตร์ 7th Hyogo Prefectural Shunda Shungun Senior High School. Okay. 8th Pranarai School. 9th Princess Julapon Science High School นครศรีธรรมราช 10th National Institute of Technology Kosen Fukushima College. To begin the sessions, we first like to explain the guideline of each presentation. The time allocated for each presentation is 15 minutes, 1 minute for preparation, 7 minutes for presentation, and 5 minutes for commentators to comment and discussion. Another 2 minutes for certificate and take photos at the end of each presentation. For additional information about certificate, today you will receive only a cover of the certificate, and you can download the certificate on the website TJSSF 2023 later. We apologize for your inconvenience. 1 minute before presentation time's up, we will show you this sign. Yeah. And when the presentation time is up, we will show you this sign, which means you have to end the presentation. Please mind the time shown on the monitor. Today's session is being recorded for viewing as post-conference, or you can watch the live stream on TJSSF 2023 channel. During the presentation, please turn your mobile phone on silent mode and please keep quiet as much as possible. If you want to take photos, please stay in your seat and refrain. For additional information, we will start the timer when you stand in front of these computers. เอาเลยวะ Is ผาแดง วิทยาลัย school here อะไรนะครับประเด็ง yes ขอโทษครับ okay what about Princess จุฬาภรณ์ นครศรีธรรมราช okay Ladies and gentlemen, without further ado, it's my pleasure to welcome our first group from Princess Chulaporn Zion High School Lei with the project entitled The Study and Isolation of Indigenous Bacteria that Produce Amylase Enzyme from Soil Surrounding a Tapioca Flour Factory for development as Bacterial Prototype in Wastewater Treatment. Please give them a round of applause.
My name is Pumipat Kamchumpu.
And my name is Gekong Khunpon.
We are from Princess Chulaporn Science High School. Lei. Today, we will be proud to present the study and isolation of indigenous bacteria that produce amylase enzyme from soil surrounding a tapioca flour factory for development as a bacterial prototype in wastewater treatment. For the first, nowadays, instant tapioca is a common ingredient in many cuisines, including desserts and bakeries. Instant tapioca products are significant in Thailand and are also exported as an industrial product from Lei province to both domestic and international markets. During the production process of the instant tapioca flour, approximately 10 to 20 cubic meters of wastewater is generated per 1,000 kilograms of tapioca. However, the existing factory of system for managing this wastewater are inefficient, leading to problem of foul smelling and widespread water pollution. This had adverse effect on the livelihood of people living in nearby around the communities and schools. This bacteria will be developed as the bacterial amylase prototype for the treatment of wastewater. This approach aims to enhance the efficiency of industrial wastewater treatment system, making it a safe and biologically treated water suitable for discharge into public water sources. For the objective to develop the effective bacterial amylase starter on wastewater treatment from tapioca flour factories. For the variable, the independent variable is bacterial isolate from soil near tapioca flour factory. The dependent variable is efficiency of bacteria in treating starch wastewater. The controlled variable is amount of bacteria, nutrient media, and temperature and time. We can divide it into three steps. The first step is isolation of indigenous bacteria from soil. First, take the soil sample, and second, dilute the soil sample with tenfold dilution. Third, spray plate on nutrient agar. The fourth is isolate the pure culture bacteria, and the next step is caustic with a single colony.
In step 2, selection of bacteria capable of producing amylase. First, we start from polyinoculation pure bacteria on start agar and pour with 0.5% iodine solution to test start and observe for the clear zone. Then measuring the length of clear zone diameter and colony diameter to calculate hydrolysis capacity. Step 3. Testing the efficiency of starch digestion in synthetic wastewater. We start from being the efficiency of bacteria from the step 2 in nutrient bath and compare turbidity with 0.5 McFarland standard with by UV visible septophotometer and incubate bacteria in synthetic wastewater and check for percent by alcohol reflectometer every 3, 6, 12 hours. For the conclusion, a total of 16 isolates were obtained from the soil. Among them, 10 isolates demonstrate the ability to digest starch dot amylase productions. Among these, KP08 exhibit the high 8 efficiency with high 8 hydrolysis capacity at 2.317, followed by TD01, KP06, KP10, and TD03 respectively. The efficiency of starch digestion was tested in synthetic wastewater by incubate in candle jar for 7 days. The results show a maximum increasing sugar content of 4.4% big, with a tendency to increase continuously. Thank you for your attention. If you have.
Thank you for your excellent presentation. May I invite our commentators to give some comments to the presenter, please?
Okay, your content is relevant and your format is visually appealing, and I can follow your work with, I enjoy listening to your presentation. Well done. Okay, so I just only one comment on your presentation on the isolate you work with. Can you specify or identify the species of bacteria you work with? For example, the isolate KP08. What's the names? What's the species name or genus of this isolate? Can you give the name of
We can identify the bacteria for the species, but we can classify on the basic morphology and with dried gram stain. Yeah. For classify only the bacillus for SPP. Yes.
Okay, I have just a few questions before suggestion. Before make a suggestion, I have just a few questions. How you isolate your bacteria from the wastewater? At the first,
We isolate the bacteria from the soil around Tabelga Fowl Factory, not in water or wastewater.
yes, and in your lecture, I see you make a dilution, right?
Yes.
Before culture bacteria.
Yes.
Okay. Which dilution does you choose to spread or stay, or whatever you do like that. Can you remember? Which dilution you choose to use to spread?
We use dilution in the 10 raised to power 4.
Oh, okay. Minus 4?
Yes,
10 to time to minus 4. Okay. Actually, I think your research, your study is very benefit to use and to apply to industrial scale in the future. Okay. But I think if you should another way to evaluate your test, now you just use a percent brick to test which bacteria, which isolate is efficiency, right? If you use another test to better to evaluate, okay, you can review its first, okay? Maybe it can make your study that's have impact, more impact, okay?
Thank you.
Thank you for your constructive comment, commentators. Does anyone have question about this project? No? Okay. May I invite Dr. Shontisha Jinaporn to award the certificate to our presenter in front of this black dot, please. Excuse me, Professor. Thank you for both commentators and presenters. And the next group will be present at 9:45.
that, we analyzed the feeder crab behavior by record a video clip. And next, we analyzed soil quality and microplastic in soil. Analyzed the microplastic in the feeder crab. And after that, we analyzed the statistical data. And for soil quality measurement, we study about 3 important aspects, which are pH, NPK, and organic matters. And procedure for microplastic analysis, we first sieve the soil with a sieve of size 5 mm and 1 mm. After that, weigh and bake. We use our chemical, ferrous sulfate and hydrogen peroxide to digest the soil. And after that, filtrate and bake and observe under the stereo microscope and do the same process in the feeder crab, but we use hydrogen peroxide and potassium hydroxide for the chemical. And this is methodology for observe the behavior of feeder crab. We record a video clip by 7 minutes each from 15 samples of each crab type. Study their behavior as follows: feeding, walking, flirtation, burrowing, hiding, fighting, cleaning, and standing.
Next is the results part. First is soil quality study. In soil quality study, we show that the pH and the pH and the NPK in Ban Butanoi area has greater number than the salty hot spring area, but contrast with the organic matters. Next is the study on the microplastic in the feeder crab and also in the topsoils. We see the same, like in Ban Butanoi area has greater the amount of microplastic than the salty hot spring area, and the common size of the microplastic is between 1 millimeter to 300 micrometers. Next is the number of microplastic in the feeder crab, and we found that in female, there detects more microplastic in female and the big one, yeah. Next is the characteristic of microplastic that we found. We found only 2 types of microplastic, which is fragment and found fiber the most. And this is the example of microplastic that we found. And this is the color of microplastic that we found. We found many colors of it. It's clear, white, red, black, green, and blue. And this is the example of microplastic. Next one is the diversity of feeder crab behavior. In the figure 3, it shows the behavior average behavior changing in 7 minutes. It can be seen like in salty hot spring area has more time to has more average higher than the feeder crab in Ban Butanoi area. And the next one is show the feeding rate of the feeder crab. You will see like the feeder crab in salty hot spring area has a faster feeding rate than the feeder crab in Ban Butanoi area. And the female will faster than the male because female has 2 feeding claws. And this is the original claw of the feeder crab we will have only in male feeder crab, and we don't see the difference between this study. And this is a study on the behavior of feeder crab. First is the behavior group that we found in male feeder crab only, which is flirtation and fighting. And flirtation and fighting in both of these, the feeder crab in Ban Butanoi area has greater time to do this behavior than the salty hot spring feeder crabs. And next is the behavior group that we wouldn't found in Ban Butanoi area, which is hiding, burrowing, and cleaning. Next one is behavior group that we found in both study sites, which is feeding, standing, and walking. In feeding and standing, we found the same, like in Ban Butanoi area has time to feeding and standing more than the salty hot spring feeder crab, contrast with the walking behaviors. And in conclusion, microplastic in the soil and also in the feeder crab in Ban Butanoi area has a greater the amount of microplastic than the salty hot spring area. And the discovered, the common is fiber shape and with the color blue, green, black, red, and white. And this maybe come from a fishing net. And the microplastic in the feeder crab, the more microplastic detects in female and the bigger one. We found the microplastic in the older or the bigger one because it has a longer time to expose the microplastic. And the last one is the feeder crab behavior. The microplastic may be enter to the feeder crab and affect their metabolism. From the result that show that Bamotanoid area has show less active behavior compared to the feeder crab in salty hot spring area, which actively and faster. After we finish our project, we come to the community to talk to the children, like their school in the community, to make them know that their plastic waste have impact to their community, and the microplastic have effect to their feeder crab, and make them know how to check it and how to avoid this one. And we would like to thank to our all supporter. And this is our reference. Thank you for listening.
You already have the natural ability to hand out the presentation, and very well organized presentation.
Thank
Very
you.
nice, well done. Okay.
Thank you.
I have a few comments on your academic research. Please go to page 20. Okay. In this result, you measure the behavior of the feeder crab
Yes.
within 7 minutes.
Yes.
So I want to know the reasonability. Why do you focus on only 7 minutes for measuring the behavior or observing behavior of them?
This is our first research, and I think 7 minutes is the best period of time that we can see the behavior of feeder crab. Like, it's changing a lot in 7 minutes. So I think it's the best period of time that we study by record a video clip for 7 minutes.
What will happen?
It's because of in 7 minutes it will has the different, because if we study more in 14 minutes, we will see like there's do their behavior in like equally. Like in 7 minutes, there are some feeder crab that didn't do anything. But if it's more time, it will be, oh, I need to do this, but a little. But the 7 minute is there has a past research about their maybe, I don't know, hermit crab, yeah, and they're like survey only 7 minutes too. That's why our research used this time, yeah.
Okay. And how can you control the environment around them when you're observing them? Because you work with 2 different habitats, right? Yeah, Ban
Bamotanoid,
Hanoi and salty hot spring. Okay.
Is it the
Because
same?
of temperature or other
Yeah.
environment factor might affect their behavior.
Yeah, also the weather was affect too, but we used to study site with difference in salty hot spring and Bamotanoid area. It's because of it was the same sea, same ocean, same coastal line in Trang, yeah. And this place can we see the different clearly, because in Bamotanoid area, when we come to this place, we see a lot of plastic waste. But contrast with the salty hot spring was the place that we keep their community keep them to be avoid of tourists, and no one wants to be there. Yeah.
And in my opinion, I very agree with Ajahn Wuttipong, your presentation is very crystal clear.
Thank you.
Thank you so much.
Thank you.
Thank you, Professor. May I invite Dr. Wuttipong Mahakam to award the certificate to our presenter in front of this backdrop, please. And the next presenter from Furukawa Remer Junior and Senior High School with the project entitled Methane Fermentation with Hot Spring Water. Please give them a big hands.
Sawadika. Hi everyone, we are Furukawa Reimei Junior and Senior High School students. We are from Miyagi Prefecture, Japan. We are going to tell you our research. Our research is methane fermentation with hot spring water. In Miyagi Prefecture, Naruko Onsen is part of Osaki City. We live in there. There are hot springs and hotels. Also, discharge of water in Naruko Dam in summer and red reefs in autumn are famous. A lot of people visit Naruko to watch them, but visitors have been decreasing after COVID-19. We want to increase visitors more, so we focused on the methane fermentation using hot spring as a heat source and food waste from hotels in our city. Our objective is to make a safe and renewable cycle for methane production while reducing carbon emissions. First, what is methane fermentation? It's a process that produces methane gas. Methane bacteria make food waste into methane gas and liquid fertilizer. Methane is vulnerable, so we can use it to boil water.
In Japan, food waste usually is burned and may release carbon dioxide. Methane gas, burning methane gas, it released carbon dioxide, but this methane gas is from food waste, so this activity is carbon zero or net zero or carbon neutral. In our experiment, we used methane fermentation machine, food waste such as vegetable and fruits. Also, we used microbial community such as methane producing bacteria. Methane fermentation has good things and bad things. Good thing is vegetable, fruits, and stems. Bad thing is meats, fish, and rotten vegetables. That's because these bad things doesn't work better. This is method. First, we put 400 milliliters of food waste in the blender and smash. Second, put them with methanogen, and methanogen is microbial community. Yeah, like this. Step 3, fermentation time. So we get 20 milliliters of liquid fertilizer and methane gas. Step 5, put food waste into the machine regularly and return to step 3. Yeah, regularly. We want to methane fermentation has two types. One is middle temperature fermentation around 30 degrees. Another is high temperature fermentation around 55 degrees. So we want to know how does the generation of methane gas speed change by temperature. Oh, this is our result. In 30 degree and 55 degree. In 30 degree, it took 15 days to fill gas back, but in 55 degree, it took only 3 days. 3 days. Oh, so 55 degree is more suitable than 30 degree. This is our conclusion. From our result, 55 degree is suitable temperature for methane fermentation. So we want to heating by electricity. We would consume 38,815 kilowatt hour. It means we would release about 1.4 tons CO2. Yeah. But by heating by hot spring water, we do not need to use electricity, and methane could be used to boil water and cooking snacks, so we could reduce the amount of fossil fuels.
Next, we want to experiment with this machine. This is methane fermentation machine. It can be done all methods by itself. We would like to experiment to see what would happen if we heated it with hot spring water and added food waste from hotels. And we want to make a business model with methane fermentation and used hot spring. First, we collect food waste from local hotels in our city. Second, we start methane fermentation with food waste and hot spring water as a source of heat. Then we sell the liquid fertilizer to farmers. We want to provide something to cook by using methane gas. Finally, food waste and crop waste are collected, allowing us to start the cycle again, so people enjoy our city more and more. We want to increase in tourists in Naruko. This is our reference. Thank you for listening.
Okay, you both are effective researcher, but I think you tend to be a little shy when you're presenting your research. Okay. I like the visual graphic you use in the presentation, but it would be better if you can increase the size of the figure or the font size to make it bigger. That's better. Okay. So, okay, great job. Okay. I have only one question concerning with the research you work with. How can you trap the methane after producing by the bacterial community? Trap the methane for longer use. Methane gas. Keep it for longer use. Keep the methane gas for longer use after you produce.
To use business model, we think like balloon and balloon. This machine and this machine connect gas bag.
Excuse me, this is your fermenter, like a fermenter.
Yeah.
Okay, and you use you push the fertilizer in there, and some material, whatever you want to fermentation in there. Okay, and last product is how to keep it to use in another process. How you keep it? How you harvest the methane from this fermenter? You are from your machine. After fermentation, the methane is occur, right?
Yeah.
Okay, and what after that?
We think a liquid fertilizer cells put bottles, liquid fertilizer so bottle put in bottles and sell for local farmers.
Okay, I just have a few questions too. In your experiment, it seems like you use two condition temperature, right? It's 55 degrees Celsius and 30, right?
Yes,
Can I see? Okay, and which one is better? Which one is better condition from your result? Which one is good one? 55. Okay. I think is if it's possible, you should add more condition to study in the same time. Okay. And can I ask you how to prefer your fermentation? How to you prefer your fertilizer to use as a starter? How to prefer? How you shoot it to be a starter?
どうしたいかってことどうしたいかもっとだから増やしたい
In your fermentation condition, you add fertilizer, right? How to shoot it? How proof is this a good one?
Excuse me, sorry to interrupt. Our time is up. Thank you to our
That's
commentators
fine,
and the
don't worry.
presenters. May I invite Dr. Shuntisha Jinapon to award the certificate to our presenters in front of this backdrop, please. And the next presenters from Pathum Vilai School with the project entitled A Comparative on the Efficiency of the Ionizer and UVC Generators in Reducing the Reproduction of Bacteria in Pathum Vilai School Cafeteria Cutlery. If you're ready, you can start.
Okay, ครับ test okay. Good morning everyone. My name is ทรงโปรดรักษภักดี
And my name is Sethisa Thanashawan.
Our project's name is comparison of the efficiency of ionizer and UVC dryer in reducing bacteria multiplication on cutlery in Pathum Vilai School canteen. Terminology: ionizer refer to machine that can create negative ion. Bacteria refer to bacteria found on cutlery in a specific sample cutlery area of Pathum Vilai School. Petri dish refer to petri dish that use potato, dextrose, agar as food for growing bacteria. Efficiency refer to ability in reduced number of bacteria. Introduction. Our project begins with cutlery. As you can see in the canteen or maybe food court in Thailand, whenever you pick your spoon or fork, you're gonna have to stir it in this boiling water. But the health department state that this boiling water is not clean because general temperature of this water is around 50 to 60 degrees Celsius, so that it's not hot enough to clear all germs that appear on the surface of the cutlery. And the resolution of this problem, we have a UV drying machine and ionizer. So our project objective is to compare these to machine in the aspect of reduce amounts of bacteria that can be found in the surface of cut relief.
The next is material and methods. Part 1, we designing the ionizer. Step 1, we design a model of ionizer with corridor program. And step 2, take an acrylic and cut it with laser cutter as designed. And last one, step 3, take each piece of acrylic and assemble it together with liquid binder. In the yellow area is area for bringing peptides and cutlery, and the green area is we install terminal and ion generator. Okay. The results from the experiment were collected 3 times in total to ensure the accuracy of the data.
Results and discussion. This graph shows the different potential that occur inside the ionizer. So as you can see the graph, we have 2 phases, that is ionizer is on and ionizer is off. And you notice that the different potential inside the ionizer is in the range of minus 26.13. So in this we can conclude that the ionizer can create the negative ion. And this second experiment we described showed about the efficiency against germs on cut relief. As you can see, we compared about average performance of ionizer and UVC light. And you will see the UVC light is most effective in this experiment.
This graph is testing efficiency against each type of bacteria on a petri dish. It was found that of bacteria type, It was found that the type of bacteria present on the petri dish does not affect the ionizer and UVC dyer, but its efficiency of ionizer decreases when the comparing of the ionizer and UVC dyer. And last one is conclusions. The conclusion is from the result of experiment, it was found that the potential difference between the ionizer had greater effect on reducing the number of bacteria on culturing than the petri dish, because the negative ion were unable to reach the bacteria on the culture medium. As a result, the efficiency in reducing the number of bacteria decreased. Therefore, to reduce the proliferation of bacteria with an ionizer, bacteria should be placed in area where negative ion can be easily accessed, such as on cutlery. Thank you.
All right, your work is fantastic. Okay, your speak is clear and concise manner. Okay, I'm very impressed with your presentation. For the question related with your research, in the first presentation, in the first slide, you mentioned about the ionizer. You give the definition of the ionizer about it can provide negative ions. So I'm curious to know, how can it work with bacteria? How can
the negative ion affect to the bacteria?
Yeah.
Because the negative ion can pull out the hydrogen inside the bacteria. So it's affect in bacteria.
Hydrogen?
Sorry?
You mean around the hydrogen? Pardon?
Inside
Inside
the cell bacteria.
Inside.
Yes.
And when it go inside into the bacterial cell,
It will pull out the hydrogen inside the bacteria out, so it cannot like grow more.
okay. So for the cell wall of the bacteria, do you know Which ion?
Which
That-
ion?
That around the bacterial cell wall.
This
Which ion that form around the bacterial cell wall? You know?
we maybe not.
We only know this negative ion can bringing hydrogen of cell bacteria. Yes.
Okay, I have, I also have just a few questions. In your condition to study, how long you use to test this? Because you not yet tell us enough for the condition. How long you use the ionizer to kill bacteria? How long? In your test?
We test many times, but I think we take 100 times.
How long is how minute? Yeah,
1 minute. Yeah.
just
Yes.
1 minute. Why you choose just 1 minute to test?
Because it's maybe it's a good time. Maybe I mean the not too long and not too like yeah.
Or have a previous study before you test this, and they just 1 minute is enough to kill a bacteria.
We cannot infer that, but according to the experiment, we can say that it can.
Okay. So in your result is not yet to kill 100% of bacteria, right? So I would like to give us just a suggestion to you. If you add more time, it's longer than just 1 minute. Maybe you use 2 minutes, 5 minutes, 10 minutes. Maybe it's very efficiency to kill a bacteria. Okay, and how you evaluate your ionizer is can reduce a number of bacteria. How you evaluate?
We use colony count, colony digital counter. Yes.
Okay. Before and after use the ionizer, you compare it.
Yes. Thank you so much.
Thank you.
Thank you, commentators. May I invite Dr. Wuttipong Mahakam to award the certificate to our presenter in front of this black dog. The next group will be presented at 10:30. And if you want to go to toilet, toilets are on the right side of the first and second floor, and also on the left side of this second floor. And drinking water and snack are also served on the table beside the classroom number 2301. And the next presenter from Padang Vitaya with a project entitled Inhibition of Black Mold on Mango Leaves with Trichoderma fungi. Please give them a round of applause.
Hello everyone, my name is Chanyala Krupan.
My name is Jenny Pachu. We are from Pradeng Vidya School. Today we are going to present about project that name Inhibition of Black Mold on Leaf Trichoderma fungi. Well, we appreciate to show you. At the present time, mango is considered an important cash crop that is in great demand in the market. Some farmers use chemical to kill insects, but it doesn't think about consumer safety. The project organizer day for two of leading the way about Trichoderma sinum to inhibit Meliola species.
For our objective, 1. To shout out the Mariola species fungus for mango leaf with crisis black mold. 2. To study the cause of the Trichoderma sinum fungus in potato dactylus agar culture medium. 3. To study the efficacy of Trichoderma sinum fungus in inhibiting Mariola species fungus in the laboratory. For our objective, we study T-PASS.
Part 1. Study of mold separation Meliola species from mango leaves. The experimental methods. First, look at the mango leaf with mold by microscope and then cut it to a size of 1 by 1 centimeter squared and then press them in Clorox with concentration of 5% and let them stand for 1 minute and then rinse to time with distilled water for 1 minute. And the last one is placed on potato dactylus agar culture medium, incubate at 30 Celsius degree for 3 to 5 days. Experimental results. From the sorting of black mold via mango leaves, the morphological characteristics of Meliola species are velvety initially. It was cream to color. Later, the color darkens and become blackish brown. The morphological characteristics of the fungus under the microscope are synthetic fiber, which have a spherical, almost cylindrical shape and have a septum. The fiber contain chlamydospores that are transparent and colorless.
Part 2. Study on the cause of the Trichoderma sinum in culture medium potato dactylus agar. Experimental methods. We add the Trichoderma sinum livening agent to the potato dactylus agar culture medium is an amount 0.1 gram. Spread the infection theory. As incubates at room temperature 30 degrees 40 to 7 days. And check the presence of dietary fiber in foods and apply the appropriate data key to classify mold in the air. Experimental result. External morphological characteristics. First phase, the fibers are colorless. Later they turn olive green. Characteristics of fungi under the microscope. The cornea area is green and fluffy. The underside of the cornea is colorless. The cornea have a smooth surface. The chlamydophores buds in a triangular shape. The villi is thin and elongated. The chlamydospores are in the fiber.
Part 3. Study the efficacy of Trichoderma sinum to inhibit Meliola species fungus. Experimental method. First, the Trichoderma sinum fungus is spine to dissolve spore. Take it to a dilution. Cell concentration is 7.05 by 10 to the power 5 cell per milliliter. And then take it to a dilution 10 times in increment until the cell concentration is 7.05 by 10 cell per milliliter. And then later is mixed in potato dactylus agar culture medium. And when the culture medium was hardened, cut off the Meliola species fungi with cork borer and press them in and press it on different concentration of Trichoderma sinum. Incubated at 30 Celsius degree and recorded every 24 hours. The experimental results were recorded and the percentage inhibition of fungal growth was calculated using the following in this formula.
Experimental results. Table 3.1 show the efficacy of Trichodoma senum to inhibit Meliora species fungus in the laboratory. We did it 6 times every 24 hours.
But table 3.2 show percentage of mode inhibition Meliola species using Trichodoma hansenum to inhibit mode at various concentration in the laboratory. If you see in the table, you can see the base mode inhibiting agent in 96 hours at a concentration of 7.05 by 10 to the power 4 cell per milliliter. And at a concentration of 7.05 by 10 cell per milliliter can inhibit mode at least 144 hours. Thank you for listening our project.
Okay, your presentation is very clear. Okay, and a nice presentation.
Thank you.
But something that I want to mention about the presentation, the graphic is very clear, graphic is very beautiful, but only one point that I want you to concern about it is the scientific name you write down on the slide. For the scientific name, it must be italic. Okay, so you show not italic. Okay, because it's very important to know that. Okay, for the question that I concerned with is for the real application, how can you apply the trichodoma onto the mango fruit to control the mold? Because you work with in the laboratory, you isolate the mold from the mango fruit wall and then apply onto the agar plate. But for the real application, how can we use the trichodoma to control onto the mango's fruit?
Okay, we use 50 trichodoma gram, 50 gram trichodoma, and 20 liters of water, and we pour on the land under the tree or spray onto the waters. Yes.
Okay. In my question, I just want to ask you about why in your study, why you have to use 5 Clorox to soak a mango leaf before you isolate a fungi. Why?
Because the 5% and is good for this for and I think is very perfect. Okay.
In your opinion, a 5 Clorox is have an effect or not to kill microbial in your sample?
Yes.
Okay. So in my opinion, if I do the test by myself, I will not soak it in the Clorox. I will directly to isolate from mango leaf. Maybe you will have you will find many fungi, okay, many type of fungi or bacteria, more than just one. So the next question to how you identify the fungi to Meilola?
Meliola.
Meilola, okay. How to identify it? How you know that fungi is Meilola? How to know?
Because we find the mango leaf with mold, and we cut it to a size of 1 by 1, and we saw.
Okay,
observe too.
The spore shape or separate or something like that.
Yes, and we have information in school.
You compare it with the guidebook.
Yes, yes.
Okay, thank you. That's good. Okay, okay, thank you so much.
Thank you.
Thank you, commentators and presenters. May I invite Dr. Shontisha Jinaporn to award the certificate in front of this backdrop, please. And the next group will be present at 10:45. The next presenter is from Demonstration School of Khon Kaen University Secondary Siksasat with a project entitled Biodegradation of Palm for Sugar, Biodegradation Cellulose Microorganism to Hydrogens. Please welcome.
Ohayou gozaimasu, minasan. Good morning, everyone. Our project is called Biodegradation of palm fronds for sugar by degradation cellulose microorganism to hydrogen. My name is Panpanida Vacharasiri Banlu.
My name is Anwadi Jamroon.
So, palm fronds, right? When oil palm fronds reach their maturity for harvesting fresh palm fruits, there is a necessity to remove the palm fronds, which will result in a lot of organic waste, and thus it is essential to carry out the disposal. The discarded fronds have something that's called lignocellulose and lignin. Lignocellulose is popularly utilized as a raw material for producing renewable energy, such as hydrogen gas. Microorganisms capable of degrading hydrogen gas, cellulose, are used to break down the lignocellulose-rich palm fronds, and then hydrogen-producing microorganisms are introduced to break down to ferment the broken down cellulose and generate hydrogen gas. Our objective is to study the optimal duration and quantity of palm fronds for cellulose degradation in order to produce sugar using cellulolytic microbial consortium, and to select the suitable experimental sets using palm fronds for the production of hydrogen gas. Our hypothesis is that if the duration and quantity of palm fronds have an impact on the sugar production by microorganisms to generate hydrogen gas, then the longer duration and the larger quantities of palm fronds would enable microorganisms to produce more sugar and thus lead to more hydrogen.
Next is variables. Independent variables are the duration of cellulose degrading and the quantity of palm fronds used. Dependent variables is the amount of generated sugar and the quantity of hydrogen gas produced. Control variables are the type and quantity of microbial nutrient, the environment for the microorganism, the palm type, the temperature for microbial cultivation, and the presence of other gases in the bottles. Scope of study: 1. The palm fronds studied were obtained from the same oil palm tree plantation. 2. The culture nutrient used is peptone solution. 3. The temperature used for cultivating the microbial culture is 34 degrees Celsius. Next is experimental procedure. First, put palm fronds in the bottles. 2. Start the fermentation process. 3. Analyze the gas generated each day using a gas analyzer. 4. Analyze our experimental sets for sugar using a spectrophotometer and comparing it with the standard graph of sugar DNS test. 5. Select the top 2 experimental sets that can produce the highest concentration of sugar and inoculate them with a hydrogen gas producing culture at 20% of working volume of the substance. Collect biomass gas generated each day for analysis of produced hydrogen gas.
And next is the results. As you can see here, the sets using 50 grams per liter of palm fronds at the 3-day duration produce the highest concentration of sugar at 0.570 grams per liter. And the second highest is 10 grams per liter at the 3-day duration. That produced 0.491 grams per liter of sugar. And then, so we chose the 2 experimental sets and introduced them into hydrogen producer. And then we collect the gas until it stopped producing any more gases. And then we found that at 10 grams per liter, it produced 54.4 milligrams of hydrogen. And at 50 grams per liter, it produced 14 milligrams of hydrogen. So we come to a conclusion that the palm-fronds ratio of 50 grams per liter at a 3-day duration can produce the highest concentration of sugar. The duration and quantity of palm fronds have an impact on the sugar production by microorganism, which is used to generate hydrogen gas. Similarly, the palm fronds ratio of 10 grams per liter at a 3-day duration can yield the maximum production of hydrogen gas. Thank you for listening to our presentation.
Okay, thank you for such a clear and well-structured presentation. I'm very impressed with the presentation. It makes me think about the innovative research. Okay, so for the question, I want to know about the reason that why you choose the palm frond to be substrate material in this research. Why don't you choose other plants?
So basically, to put it simply, my friend over here has a palm, a place with a lot of palms near her house. And so we decided that why not use the fronds that were left there, yes. Yeah.
Can I take a look your last table in this hall, in the list hall? Okay. In this table, in your conclusion, which condition that you that you decided is the best condition?
Right. 10 grams per liter at a 3-day duration is the best condition.
In 10 or 50?
10.
Oh, it's 10, okay.
Yes.
Because I agree with you because it seems like it's not different in the product. Okay. And in your opinion, after the next step to study, if you have a time or have something whatever to make a research, okay, what do you do with your next step to the research?
My next step to research,
if you need to do.
Right. So the hydrogen that we produce, right, can be made into renewable energy, which can be like fuel and stuff like that. So maybe if we get the chance to continue our research, we will try to use these hydrogen and convert them into renewable energy. Yes.
Okay, one more question. I want to know which bacterial strain help produce hydrogen gas in your project?
Pardon me, can you repeat the question, please?
Okay. I want to know the bacterial name, bacterial isolate you use to help produce biogas here.
So it's called Clostridium, but Clostridium.
Clostridium.
Yes.
That's important. You can specify this name in the methodology.
Okay. But it has many kind of Clostridium, so we don't really know which one is specifically used for this process. But it has gone through the enrichment that we guarantee that.
Excuse me,
Yes.
if you told us you use Clostridium in your experiment,
Yes.
which the species of Clostridium? Sorry.
Yes, I was gonna. Okay, so there are a lot of species in that Clostridium, but we don't really know which one it was.
Okay, Clostridium species,
yes.
because of I think I a little bit worry about that bacteria, because it's pathogen, you know, that can cause of disease.
Yes,
So
yes.
you should very very uh, carry when you use. Okay.
Okay.
Okay. Thank you so much.
Thank you for commentators and presenters. May I invite Dr. Wuttipong Mahakam to award the certificate to our presenter, please. The next group will be present at 11. During this time, if you're thirsty, you can get the drinking water beside of the classroom number 2301.
Hello, we are Sanshokan High School student. We research about the behavior of uptime Ritalina radiator. Uptime Ritalina radiator is a type of sea snail belonging to the little ready. These snails were known to avoid seawater. Look at this picture. They live in a splash zone that is above high tide line and that is very dry. Also, if we put them into the case with a little seawater, they avoid it and go up in the case. We wonder why these such as sea snails that living in the sea area is avoid seawater and living in the dry area. And to determine the reason and mechanism why the irradiator avoids seawater and live in the dry area, we conducted one study site and 4 experiments. Firstly, we conducted study site at the Kosen Beach at Nishinomiya City, Hyogo Prefecture. The method is this. Firstly, we set up 4 sections every 5 meters from a south edge at a high tide. And we counted the number of irradiator by using the quadrates. This bar chart is a result. The horizontal axis expresses the sections and the vertical axis is the population density. From this graph, we can find out that the far from a south edge, the more population density is. Also, from this picture, we can find out that the far from a south edge, the more deeper the sea level is. For these things, we guess that irradiator needs seawater to live, although they avoid seawater. And secondly, we conducted experiment 1 to determine the reason why the irradiator avoids seawater. The hypothesis is this. Generally, it is said that bottom of the living area of sea snail is determined by the biological factors such as predators. So we guess that irradiator avoids seawater to escape from predators. To demonstrate it, we conducted experiment 1. In the experiment 1, we used this T. clevier that is considered as a predator in sea. And the method is this. First, we set out 10 test tubes filled with the same solutions. And then we put the 10 irradiator each test tubes. And we counted the number of irradiator that is above solution every 1 minute. And then we use these 5 solutions. The artificial seawater that is containing T. clevier's extract is the one that, if irradiator avoids seawater to escape from predators, it is prescribable that irradiator react to this solution more actively than the normal artificial seawater. This result, the horizontal axis is the minutes and the vertical axis is the number of systems that is above solution at that time, and pick the data at the 5 minutes and express by using the bar chart is the right one. And we can understand this graph that irradiator react to the artificial seawater that is containing T. clevier's extract more actively than the normal artificial seawater. So we guess that we concluded that irradiator avoids seawater to escape from predators. Yes, and we skip in experiment 2. Oh, sorry. And by the way, we in keeping irradiator in school, we wonder that how far irradiator can move. So to measure the ability to irradiator to move, we conducted the experiment 3. In the experiment 3, we measure the ability to irradiator move in horizontal and vertical. This is the result. From this graph, we can find out that irradiator has the ability to move more than 100 centimeters in vertical and horizontal. And in the vertical movement, in 15 or 20 irradiator remain in the seawater. This is contradicted in the experiment 1 that 7 or 10 irradiator avoid seawater. So we guess that irradiator, the behavior of irradiator is affected by the population density, and demonstrated we conducted experiment 4. The method is this. First, we set up a bottle filled with 100 milliliters seawater, and we put the various number of irradiator like 1, 5, 10, 50, 20, and so on. And we counted the number of irradiator that is above seawater every 1 minute. And this is the result. The horizontal axis is the minute, and the vertical axis is the rate of irradiator that is above solution at the time. And pick the data at the 9 minutes and express by using the bar chart is the right one. And from this graph, we can find out that the more population density, the more irradiator remains in the seawater. This result implies that they interact each other. And this is conclusion of this research. And this is the future research. Next. Future research. And firstly, in the experiment 1, the reaction of irradiator to the Kosen-Hammer speech that is living in the irradiator is less active than the normal artificial seawater. So we want to illuminate this reason, like concentration of the seawater or any other organisms in sea. And nextly, in the experiment 4, we find out that the more population density, the more irradiator remain in the seawater. So we want to research about what the component is or element is affect irradiator to remain in the seawater, like thermal or any other extractions. And thirdly, global elementary problem and irradiator that the splash zone that is living in irradiator is living area. It will narrowed. It's sea level is up due to the global warming. So in that time, how they adapt this change? And also, Suhan article suggests that the behavior of irradiator is affected by the microplastic. So we want to more consider in such a terms. And this is acknowledgement. That's all. Thank you for listening.
Okay, what I'm going to say, I love the way you present your thought and can address every aspect of your research question.
Mm-hmm.
I think your presentation is very well.
Oh.
Nice graphic, nice font, okay, and
Thank
nice
you.
speak.
Thank you.
Okay, you did a great job. So for the question, I only one point that I want to know. When you dilute the seawater,
yeah.
How can you prepare the artificial seawater, or you use the seawater from the natural seawater?
How to make the artificial seawater?
Artificial.
Artificial. In Japan, the elements of seawater is sold in any shops. It's some shops, so we buy it and put the water and make the artificial seawater.
Okay, I got it.
Okay.
Okay. First of all, your study is very interesting. Very fun to follow up your study. I very interesting about the conclusion.
Oh,
Okay, this part. You tell us the snail that they have a
yes.
communication by mucus. How to prove it? How to know?
Thank you for question. I think that in the seawater, irradiator avoid seawater often, but if the population density is high, the irradiator remain seawater. I guess the reason is if the auroral irradiator is in the seawater, they determine this environment is safe because there was less predators. So they, in that time, they will extract some of the components or elements like a pheromone and communicate each other and remain in the seawater. This is my opinion.
So the seawater can remove the mucus. You mean the seawater can remove the mucus, the snail mucus, and they cannot communicate when live in seawater.
Seawater is flow in natural, and the component is flowed. You say, okay. In the tide area, some of the places is flow in the seawater, but in the rock area, there is a small pond, like that is store the little seawater. And first place. 生息地のとこ。Yeah, there is a flooded, but in these areas, the water is not flooded because there is no flooded. So in these areas, it can be remain that irradiator extract the components or elements and remain in these areas, and they can communicate in natural. Thank you.
Just one question. In the last slide, you talking about global warming factor. Global warming. Okay. So, how global warming affect the distribution of the sea snail?
Every 1 minute to check the number of irradiator that is above seawater. Sorry, you say that I explained that we check the number of irradiator that is above seawater every 1 minute.
Every 1 meter?
1 minute.
1 minute?
Minute.
Okay. Thank you.
Okay, thank you.
Thank you for your constructive comments, commentators. May I invite the next presenter from Pranarai School with a project entitled Development of Food for Reducing Aggressive Behavior and Controlling Macaque Population in Mueang District, Lopburi Province. Please welcome.
Hello, good morning. My name is Kasawan Pusin.
My name is Chutika Najanta.
We are students at Panara School. Today we are present project about development of food for reducing aggressive behavior and controlling macaque population in Mueang District, Lopburi Province.
Background is country monkey have entered the community and are living with people, leading to various problems. Monkey have started exhibiting aggressive behavior, and the number of monkey is likely to increase continuously. This is sure have implication for the local population because the monkey population is growing each year. Macaque monkey, Macaca fasciculans. This type of monkey is found in almost every region for the regions of the country and may have group member of up to 200 individuals. They reach full size at around 3 to 4 years old and give birth to one offspring at a time. The young ones cling to the mother continuously when they are still young. The plants that are of interest for preparing foods for macaque monkey: corn, serotonin, red bean, omega-3 fatty acids, help serotonin function effectively, soybean, tryptophan. Plants of interest for study to control the productions for mammalian sperm: Alpium gas, orange, pear, neem, chloroform, papaya chloroform, tamarind flavonoids. Objective: first, to develop a complex and nutritionally balanced, ready-to-eat formula suitable for macaque monkey, which offers formats that helps reduce aggressive behavior. This will be achieved by utilizing locally available products. Second, to investigate the use for various healthful ingredients in the diet for macaque monkey for temporary contraception. This involves exploring safe, readily available, and locally affordable herbs to control fertility in macaque monkey. Hypothesis: first, using locally available products such as corn, red bean, and soybeans as ingredients in a ready-to-eat food designed to have a shape suitable for macaque monkey. This formulation aims to reduce aggressive behavior in macaque monkey by aligning with their each behavior. Second, herbs that impact prolactin production in rat-eating animal includes morning glory, sorrel seeds, papaya seeds, tamarind leaf, and tamarind seeds. These herbs induce cell death in the mammalian growth through eye test systematics. process a difference of cell death known as apoptosis. Procedure experiments 1. Production of ready-to-eat foods from a clock monkey. So, calculating foods formulas using Excel programs. And information regarding and nutritional requirements for macaca vesicularis for database NRC 2003.
Experiment 2. Studying herb affecting prolactin production in lactating animal, including morning glory, sadao seed, papaya seed, moringa leaf, and moringa seed on the result of apoptosis in mammalian sperm, preparation of experimental plants, and extraction of plant extracts. At ultra pure water filtration stored at 4 degrees and studying herb affecting mammalian sperm, detecting apoptosis by milk cell analyzer. Experiment 1 result. Proportion of ready-to-eat food from macaque monkey. Components corn 76.85%, ground meat and bone 10%, sunflower seed 9%, soybean oil 3%, salt 1%, and premix 0.15%. Result experiment 2. The proportion of the population of prolactin cells that undergo apoptosis after inhibition by the extract from Epium guerin is 99.28%. The proportion of the population cell that undergo apoptosis after inhibition by the extract from papaya seed, neem seed is 9.60%. The proportion of the population of prolactin cells that undergo apoptosis after inhibition by the extract from papaya seed is 8.08%. The proportion of the population of prolactin cells that undergo apoptosis after inhibition by the extract from neem leaf is 1.54%. The proportion of the population of prolactin cells that undergo apoptosis after inhibition by the extract from moringa seed is 0.36%. Conclusion experiment 1. The result of the study on the production of ready-to-eat food for macaque monkey, researching the nutritional requirement of long-tailed macaques from the NRC database, selecting food ingredients for use and calculating the food formula, cooking in a form that help reducing aggressive behavior in macaque from the experiment. It was found that using Excel program for formula calculation result in a nutritionally suitable food formula for every kilogram. The food consists of 768.5 grams of corn, 100 grams of ground meat and bone, 90 grams of sunflower seed husks, 30 grams of soybean oil, 10 grams of salt, and 1.5 grams of premix additive. The ready-to-eat food for macaque should have a round shape with edge porous, not exceeding 1 centimeter in diameter. Conclusion experiment 2. Researching information on herbs that inhibit apoptosis in milk fed 5 selecting herbs include Gotucola, senna seed, papaya seed, senna leaf.
Okay, due to the time is up.
Thank you very much for your presentation. Okay, I have no doubt with your communication skill, but I just want to suggest some point for the presentation slide. It's too wordy. I mean, too many words. Yes, you can reduce the word and just use the phrase when you prepare the slide for next presentation, and also make it bigger. I mean, the font is make it bigger. It's better. Okay. So for the question, I still confused about the topic with and your result present here. You introduce us with the aggressive behavior of the monkey, but when you explore the experiment, you measure the apoptosis of sperm and also the something like this. So how can you correlate the data or the relationship with sperm cell viability and the behavior of the monkey? You get my point? Yeah. Okay.
ก็คือว่าเรานะคะจะต้องแบบว่าลดปริมาณของลิงลงค่ะเพื่อที่แบบพฤติกรรมก้าวร้าวของลิงมาจากการที่ลิงมีจำนวนมากค่ะแล้วก็เราก็เลยจะลดสเปิร์มของลิงเพื่อที่แบบจะไม่ให้ลิงผสมพันธุ์กันเพื่อที่จะมีลูกออกมาเยอะค่ะแล้วก็เราก็เลยลดสเปิร์มลิงเพื่อที่จะให้จำนวนลิงลดน้อยลงค่ะแบบไม่ให้เกิดการ
Okay, so you might need to change your research topic a little bit to make it clear with audience, because some audience might confuse with the research topic you present and the result you present, because you want to control the population of monkey by reducing sperm, right? Okay, so for the sperm you used in this experiment, how can you get it? You measure the effect of your food formula on the sperm directly, right? You did not feed the food formula onto monkey directly, right? Yes. But you add the food formulation onto sperm directly. Which one? ไม่ได้ให้ลิงโดยตรงใช่ไหมไม่ให้ลิงกินเข้าไปโดยตรงใช่ไหมในงานเรา
อ๋อก็คือเราทำอาหารแล้วก็ให้ลิงกินโดยตรงเลยค่ะ
How can you collect? Okay, time's up now. Okay, that's fine. Okay.
Thank you.
Okay, may I invite Dr. Wuttipong Mahakam to award the certificate to our presenters.
Advisor is Mr. Anuruddh Misen and Special Advisor is Dr. Kesrin Rachajon. Let's start with the introduction. The date palm or Phonic dactylifera is so male and female plant are separate on each tree. It will not be together within one tree, and nowadays date palm is the crop that most Thai farmers grow more and more because it is a high yielding cash crop. So Thailand date palm are export to many countries is important economic crop of Thailand. But due to the limitations of date palm dioecious plant, so the farmer cannot determine their sex from the external observations, therefore negatively and affecting to the production efficiency and causing farmers to lose their profit. Our objective, first, to examine the sex of date palm trees by ISSR technique, to study method of DNA extraction by CTAB method, 3, to test for more primer to determine the sex of date palm.
Next to the experimental variables, the first one is independent variable are primers. For this project, we use M10 and M21. Second one is dependent variable is the sex classification accuracy percentage of date palm sex with the ISSR technique. The last one is control variable, there are species of date palms, the number of date palm sample used for examination, and DNA extraction methods. Species of date palm in experimental that we use are Konaisi, Red Bahi, Yellow Bahi, Ametahan, Jarvis, and Ganami. These are the picture of each species of date palms.
The methodology. First, we will extract the DNA from the date palm leaves from the 6 species, and this CTAB method. And this here are the sample of DNA extract from 6 species of date palm leaf. You can see the top clear part in the microtube is where is DNA resides.
Next to the second method is examining the quality of DNA by agarose gel electrophoresis technique. DNA quality was determined the absorbance by a nanodrop spectrophotometers. The absorbance of the DNA solution was measured at wavelength of 860 and 880 nanometers, and the ratio between the absorbance value was calculated. If the value is less than 1.8, there is a lot of protein in the solution as like a yellow circle. And if the value is greater than 2.0, then it's a lot of RNA in DNA solution. From the experiment, it was found that the DNA content of sample 2 was very low, therefore cannot be used for testing in the next step.
3, we will determine the appropriate primer to amplify DNA by ISSR PCR technique. And this PCR thermal cycle. 4, to amplification to the palm DNA using the ISSR marker. And this primer that we use, primer M10 and primer M21. Two of this is a base primer for ISSR technique. First, to determinations of ISSR primer specific to date. After detecting and selecting primer capable of synthesizing DNA of date palm, primer with specific yields, simultaneous results, non-polymorphic date to date were used for date palm DNA synthesis. And this blue light transilluminator used for DNA detection from agarose gel plate.
Results, agarose gel electrophoresis showing DNA bands between primer M10 and M21 in sample 1, 3, 4, 5, and 6. Experimental result 1, 3, 4, 5, and 6 are undistinguished. Summary and analysis. The analysis of experimental outcomes revealed that the selected primer M10 and M21 unfortunately did not distinct identification for
the tested samples. This outcome indicates that this primer may not possess the requisite specificity and sensitivity to effectively discern the gender of the palm tree species. We must investigate and refine the primer selection process to attain more conclusive and accurate results. These are reference primers, and these are reference. Thank you.
Thank you very much for your presentation. Okay, overall, your presentation is nice, very good. Okay, it's well and on point and cover every important aspect. Okay, but just few comments for improvement for the future work. First of all, you mentioned about the CTAP method for DNA extraction, okay? Why do you choose this method for isolating DNA? Because as far as I know, there are many DNA extraction methods. But why you focus on this methods?
Because this method is easy to do and it give more DNA results. Give an accurate result.
How can CTAP work with DNA sample? I mean, how can CTAP help isolate the DNA from plant tissues? What the function of the CTAP?
To get the DNA solution.
What is the function of CTAP?
Function.
The role of CTAP. What is the role of CTAP? Do you know? How can it work with plant tissue?
We will the CTAP method.
Yeah, the function of CTAP, because it's chemicals, right? How can it work with plant tissue? How can it help isolating the DNA from plant tissue? Okay.
First, we will cut the dead palm leaf and
That's okay. Yeah. I just want to know มันทำหน้าที่ยังไง
ภาษาไทยได้ไหมครับเหมือน CTAP method เราจะตัดใบไม้เป็นชิ้นเล็กๆแล้วก็เอามาบดในโกรง CTAP of chemical that will go to extract the DNA, RNA, and protein solution from the dead palm leaf. I don't know. Yeah.
Okay, can I have a look about the table after you use a nano drop? Okay, please describe me what about in yellow cycle? I miss your lecture.
This means it have a lot of protein in DNA solution.
And it's good or not good?
Not good.
Okay. Which one is the best product from your extension? Which one? With sample number.
Like it have to be between 1.8
Yes.
to 2.0. And-
Okay.
You use plant tissue with the same amount?
No.
No, not the same amount. Why?
Because we can find the same species, male and female in Thailand. Because in the
นี่น่าปริมาณมันต่างกันมากนะ The concentration of nucleic acid you get is variable. Very low, like for the sample number 1, and some sample, right? For example, number 3 and number 5, you got 7,000 nanograms per microliter, and for number 1, you got only 100. So it vary. So I'm curious to know the starting material, the weight of the plant tissue you use. How many grams of plant tissue you use for the extracting the DNA? How many grams?
I can't remember, but each species is เท่ากัน It is same.
Different weight, right?
No, the same weight for
Okay.
every species.
And how many? You can't remember, right?
I can't remember.
That's important, very important. Yes.
เฮ้ยแต่ว่าคอม
Have you dilute the DNA before prepare PCR master mix?
Yes. We will dilute DNA.
Now it's time's up. Thank you very much for your nice presentation.
Okay. Hello everyone, my name is Fuyuka Sano.
My name is Nao Suzuki.
My name is Momoka Kamei.
Here, we would like to briefly introduce the screening of the aspartate-producing lactic acid bacteria from traditional Japanese pickles. Amino acids have mirror image isomers called L form and D form. Living organisms mainly contain L amino acids, but in recent years, the physiological function of the amino acids have become known. Additionally, whose function of the amino acids such as the beauty effects of the aspartate, are attracting attention. Lactic acid bacteria are microorganisms indispensable for the production of fermented foods around us. In recent years, the effect of lactic acid bacteria and lactic acid fermented foods on human health have also attracted attention. Pickles are foods that give vegetables a unique flavor and preservability through the action of lactic acid bacteria. This project aimed to isolate, screen, and identify the aspartate-producing lactic acid bacteria from pickles produced in Fukushima Prefecture. Furthermore, this study is not the first such effort. Previous research includes research by Dr. Kajitani and colleagues at Nagaoka University of Technology. The experimental operation in this study was performed to reference to that study. In this study, pickles were first suspended in sterilized saline solution and spread on MRS medium and cultured anaerobically to obtain approximately 150 colonies.
Next, in order to select lactic acid bacteria from strains obtained, a catalyst test was adding hydrogen peroxide to the bacteria. And we were able to 142 lactic acid bacteria candidate strains that were confirmed to be catalyst negative, a characteristic common to lactic acid bacteria. When the aspartate concentration in the MRS culture, supernatants of all those isolated strains were measured. Only strain S39 shown at the aspartate concentration of 200 micromolar or more. However, this concentration was about 1/7 of that of the WDN19 strain, known to exhibit the highest aspartate production. As shown in the table, we also identified bacteria showing the aspartate concentration of above 100 micromolar, bacteria showing the concentration below 100 micromolar, and bacteria showing no aspartate production.
Next, partial 60S rDNA sequence homology analysis was performed on 24 strains in order to analyze differences in the aspartate production ability among lactic acid bacteria by genera and species. As a result, the A39 strain was found to be most closely related Lactobacillus sakei strain J64. Interestingly, the WDN19 strain, which has been known to exhibit the highest aspartate production ability so far, also belongs to the genus Lactobacillus. In addition, all 24 lactic acid bacteria candidate strains analyzed were lactic acid bacteria, and they were classified into the 3 types of lactic acid bacteria of the genera Lactobacillus, Lactobacillus, or Leuconostoc. All the lactic acid bacteria that produce aspartate at concentration of 100 micromolar or more were classified into the genus Lactobacillus, except for one strain, On the other hand, most of lactic acid bacteria belonging to the genus Leuconostoc, which is phylogenetically distant from Lactobacillus, has concentration of 100 micromolar or less.
In this study, we could not find any lactic acid bacteria with a higher ability than the previously reported Lactobacillus species strain WDN19. In the future, however, if lactic acid bacteria with high aspartate concentration ability are found, it will be useful for the production of fermented foods, such as yogurt containing high concentrations of the aspartate, which has beauty effects. Thank you for your attention.
Okay, I appreciate your effort to make a great presentation. Tahen Yuku Dekimashita. Okay, Sukoi. You did a great job. Okay, so for the question related with your work, I'm curious to know the application of the aspartate lactic acid bacteria. How can you apply the bacterial stain you obtain in this project for the cosmetic products or cosmetic application? Because in the introduction part, you mentioned about the cosmetic application or beauty, something like this. For the first one, no, yes, you said discovery of physiological function, for example, beauty effect of the aspartate. So after you got the bacterial strain in this study, how can you use it for the beauty effect, something like this? You know my point? You understand my point?
The aspartate has beautiful skin effect. No.
I mean, how can this bacteria help improve or help, how can I say, how can you use it for the beauty effect? By consuming or by eating or for apply on your skin, something like this?
Eating made yogurt and so on. Eating food.
You know, because you isolate the bacteria from the Japanese pickle, and you obtain them with high the aspartate activities help produce the aspartate. And I want to know the application of this strain, you know? Can we use this bacteria for skin application, like to be the component of the cosmetic product or consume, eat? Okay, that's okay. No, no, please don't be worried. Okay, about that. Okay, so let's talk about article result. Please go to the result page, result slide on the phylogenetic tree. Phylogenetic tree. On result. One more, please. Okay. One more. Next one. Okay, here. Okay, I see the strain for the first part, Lactobacillus. Okay, most of them can produce high the aspartate because you highlight with the blue one, right? But for the other strain, leuconostoc, it's produce only a little bit amount of the aspartate. All of them isolate from the same source, right? From the same pickle.
Same pickles.
So from this result, okay, time up start now. Okay.
Thank you to our presenters and commentators. May I invite Dr. Shontisha Jinaporn to award the certificate to our presenter, please. Before we end this morning sessions, may I invite all commentators and all participants in this room to take a photo together. May I invite everyone in this room to take a photo together in front of this backdrop. That's all for this morning session. Thank you to all commentators and participants. We'll see you again this afternoon. Thank you.
Is also produce the aspartate too, right? Okay, and how is made?
Lulu.
Minasan konnichiwa. Good afternoon, commentators and participants. Welcome back to the Biology 1 presentation room of the Thailand-Japan Student Science Fair 2023. My name is Pichaya Sombat.
And my name is Shawanun Pherdgel.
We are students from Princess Chulaporn Science High School Leh and the Master of Ceremony for this oral presentation. First of all, we would like to introduce our commentators in this room. The first commentator is Assistant Professor Dr. Wuttipong Mahakam from Khon Kaen University. And we also have Dr. Shontisha Jinaporn from Leh Ratchapat University as our second commentator. We are also delighted to introduce 7 schools that will be present in this afternoon session. Please raise your hand when you hear your school's name. Start with the first school, Kosen King Mongkut Institute of Technology, Lat Krabang. Pungkan School. National Institute of Technology, Kosen Hakodate College. Princess Chulaporn Science High School, Phetchaburi. Nong Bua Pithayakarn School. Sapporo Kaisei Secondary School. And the last one, Tokai University Takhanawadi Senior High School. Thank you. To begin the session, we would first like to explain the guidelines of each presentation. The time allocated for each presentation is 15 minutes, 1 minute for preparation, 7 minutes for presentation, 5 minutes for commentators to comment and discuss, another 2 minutes for certificate and take photos of the end of each presentation. For additional information, today you will receive only a cover at the certificate, and you can download the certificate on the TJSSF 2023 website later. We apologize for your inconvenience. And 1 minute before presentation time's up, we will show this sign up. And when the time's up, we will show this sign, which means you should end the presentation. Please mind the time show over the monitor over here. Today's session is being recorded for viewing as post-conference, or you can watch the live stream on TJSSF 2023 channel. During the presentation, please switch off your mobile phone or turn on the silent mode, and please keep quiet as much as possible. If you want to take photos, please stay in your seat and refrain from walking around. The presentation will start at 1:30 p.m. At this moment, please feel free and stay calm. The toilets are on the right side of the first and second floor. Drinking water and snacks are also served in front of the classroom number 2301. If you have any questions, please don't hesitate to ask us. Thank you. Everyone, without further ado, it's my pleasure to welcome our first group from Kosen King Mongkut Institute of Technology, Lat Krabang, with the project entitled The Medical Appliance for Follow-up the Respiration Efficiency of the Patients After Surgery and the Patients with Respiratory Diseases. Welcome.
Yes. Good afternoon, everyone. はじめまして。私はパイサラーแก้วสะแสนです。
私はわらしやจิตุสำราญです。高専 KMITL から来ました。
今日は私たちがプロジェクトネクネムは呼吸器疾患患者向けの医療機器です。これから私たちは英語で発表します。So our project that named the medical appellant for follow-up the respiratory efficiency for patients after surgery and patients with respiratory diseases. Currently, the medical appellant for patients with respiratory diseases cannot respond all patients, and the function is not enough for follow up the respiratory efficiency. Based on the respiratory disease mortality data screening from 2017 to 2021, we can observe that Thailand experienced death attributed to the respiratory diseases can account for 0.05% of population. However, the patient after surgery, patient with respiratory diseases, and patient COVID-19 patients need to follow up the respiratory efficiency. While spirometer is the appellant for measure the lung capacity, and the typho is the appellant for training muscle respiratory. Consequently, the medical appellant play a main part in enabling both medical professional and patient to track the respiratory efficiency, therefore contribute to analysis of the treatment in the subsequent stage. Therefore, we would like to develop the medical appellant to follow up the respiratory efficiency and training respiratory muscle within single device.
So this research aims to check and follow the efficiency of respiratory of the patient who is under the recovery treatment process after surgery. And the second is check and follow the efficiency of respiration before and after of respiratory infection patient, for example, COVID-19 patient and the affected patient by air pollution or PM2.5. The next one I will explain about the principle of this prototype. The medical apparatus consists of two main parts, which are sensing and mechanism. Regarding the principle of this prototype, the sensor component employ a counter module sensor to convert exhaled air velocity and utilize the ST11 sensor to detect humidity and temperature value. In the mechanical aspect, a spring is incorporated agreement resistance during exhalation and also respiratory muscle training. And how can we determine the results? According to the study method which use sensor and coding. So we got the data as shown on the monitoring. There are the number of breaths counted at specific time, humidity, and also temperature. And we can calculate by using airflow equation and lung capacity equation. The airflow equation used to determine the speed of exhaled air while mouth breathing. After we got the speed of exhaled air, we can calculate the volume of lung by using the lung capacity equation as shown in the slide. Result and discussion. The velocity, volume, temperature, and humidity measurement of accurate air from target patient were found to be lower compared as those of healthy people.
So for the conclusion of our project, the design that we designed the prototype can draw in the inspiration from the exciting, the appearance, medical appearance for measuring respiratory efficiency and training respiratory muscle, employing the basic mechanical and basic sensor, enhance its capability, and then this improved prototype can measure the value in the with greater precision, potential enhancing, and respiratory efficiency monitoring. So the device environment can contribute to the progress of the medical respiratory appearance. And this is our reference. So thank you for your attention.
Such brilliant presentation. May I invite our commentators to give some comments to the presenter, please?
Thank you very much for your presentations. Okay. Okay, your content is quite relevant with your research topic, but I think some part of your research is, how can I say, I see only the instrument you develop right now, but you did not measure the lung efficiency from the patient, right? Did you use this instrument with patient?
Yes, we did it.
But you did not present the data from them.
So now
how
we
many
have
people?
10 patients.
10 patients.
Yes.
Okay, can you show the result from them?
Now we didn't show in this because it's like has the low case and didn't specific like we must to know the basic information of the patient is must to contact with the hospital like in the fundamental or in the before did she or he has the basic diseases before, so we will be not sure about because a health condition about himself or herself.
So, because I can see the difference between the data before and after using this instrument in this presentation. Okay, because you mentioned you can use this instrument to help patients to monitor their respiratory efficiency. Can you give an example of just one patient that tried to use this instrument before and after and the result of them?
As we use this medical appellant which my friend that have infected COVID-19 and my friend that will be healthy people, so we can know as the speed of airflow of accurate air of the my friend that infected COVID-19 were lower than the healthy people as compared. And
so the model of this instrument is look like the asthma instrument, you know, asthma, hop hood.
Yes.
Normally, people who have asthma problem use instrument like this check their respiratory efficiency. So I don't know the model you used for developing this instrument, where it come from? Prototype? Okay.
For the prototype, we inspire from
The
these three.
spirometer.
Yes, spirometer is for measure the lung capacity, and for these two, the black is a inspiratory muscle training or IMT, and typhoid is a for training inspiratory muscle.
Okay. So okay, I got it. Your research is very interesting. It can be applied for medical treatment, medical application, can be used for the real monitoring of people health. Okay, I hope you can develop and continue working with this instrument and get some more data to present in the next future. Okay.
Thank you very much.
Thank you so much. I have a few questions. It seems like you show your in your study compare between two groups in the patient that already infected with the COVID-19 and each one group is healthy people, right? Can I ask again, you already test this or not?
Yes, already
Okay,
test.
but now you cannot put the data from the hospital.
Yes.
Okay. And what you found about it now? In the healthy people group is have a capacity to use this equipment better than the patient that infected with COVID-19 or not?
We found that healthy people have lung capacity allowed 5 or 6 liter, but in patient is lower than allowed. The data is not constant, but it lower than 5 or 6.
Okay. Can you have any table or graph for us to see? It seems like you have a data.
This data is from the experiment. Yes, this data is from the experiment, and we does not put it into the presentation slide. Yes.
So I think your study is very interesting and useful, but in your presentation, if you add more information, especially about the result, it can help the people that listen to listen to you understand more. Okay.
Thank you for your recommendation.
Now may I invite Assistant Professor Dr. Buddhipong Mahakam to give the certificate and take photos with the presenter, please. And the next project are from Bunkan School, with the project entitled The Study Morphological Characterization and Species Diversity of the Terrestrial Isopoda for Breeding in Commercial Pets. Please welcome.
Good afternoon, ladies and gentlemen. My name is Chanapon Tisat.
And my name is Glawat Manin.
So, today we would like to presentation about the study morphological characterization and species diversity of the terrestrial isopod for breeding is communication pest. The isopod have allowed 3,007 species in the world, which the most to have survey isopod discovery have been in southern of Thailand.
I'm going to start with introduction. Benefits of isopod in soil for agriculture. Disintegrate plant debris, and the isopods help aerate and soften the soil.
Not just only that, isopod is very hot in exotic market pets.
Currently in Thailand, there is no reports of isopod studies in Bangkal province, northeastern region of Thailand.
Material and methods. Step 1. Study morphological characteristic and classification of isopod in land of Bangkal province.
Step 2. Study
This is showing results according to objective 1. Morphological study determine the identify and identity species of terrestrial isopod. It was found that terrestrial isopods were classified into 8 genera and 9 species, as showing in the picture.
Step 2. Study physical, biological, and some chemical factors of habitat isopod in the ecosystem. A. Physical factor. B. Biological factor. C. Chemical factor. B. Biological factor. Isopods tend to live in areas where moisture and sunlight are difficult to reach. C. Chemical factor. Soil chemistry factors in the areas where its isopods live.
This showing resource according to objective tool as showing in this picture. Step T, guideline for breeding isopod as pet.
Guideline for reading isopod at pest. There are steps as follow.
1. Take isopod of the same type in a four latest box.
2. Simulate test the environment in which the isopod species live, such as bugs, moss, and soil types.
T. Temperature and humidity control. Moreover, it also adds a springtail creature to clean up the beet eaten by isopod. The isopod propagation takes 3 weeks. This brings me off the end of the presentation. Thank you for your attention.
Thank you very much for your presentation. Okay, you tend to be excited, right?
Yes, I'm very excited.
Okay.
It's my first time of the presentation.
Okay, all right, no problem. Okay, so for the question related with your research, can you identify the species of isopod you work with? Species name.
Oh, species name. We found just 9 species.
9 species.
Yes.
Which guideline, which textbook, which paper or document that you use to identify them?
I use key in internet because in Thailand
From
don't
internet?
Yes.
Oh, is reliable?
Again, please.
I mean, is it reliable? I mean, sometimes when we search the information from internet, some data source is not reliable because we
yes.
need some document that have some detail, right? Morphological description, figure, some drawing, and dichotomous key, something like this, to help identify them.
Yes.
So you make it like this?
I use key in internet because in Thailand don't have many to survey about isopod.
So you got the name of them from internet, right?
Yes, I got the name in.
I actually
I have a name too. But
so can you show the name of them?
Yes,
here?
sure.
Yeah.
This is a name of isopod.
No.
Hey.
So, no. Internet sources. Do you have the paper or research paper that support the data?
Yes, I have, but it in the home, and I have a real one of isopod in my room.
Maybe you said by the data from the internet is mean paper. It's not just Google, right?
Yes.
Okay. Do you have the document, research document, and compare the data from your project with that?
I don't have now. I don't have it now.
Okay, so for the next one, I want to know which one suitable for using as a commercial pet from your opinion.
Oh, it's I breeding just classification of isopod and morphological characteristic forbidding to use combination paste, but now it a small project. I will continue this project more.
Nowadays, some people pay attention to use this isopod to be pet, right?
Yes.
Which one that, which characteristic that they like to use as a commercial pet?
Is have many species to use to combination pet.
I mean, which one is popular?
Oh, popular is not in this picture. In this picture is we found. We found in real one, but is popular. It's called duck.
Pardon?
Duck.
Dark one.
Duck. Is isopod popular in Thailand. It's called duck. D-U-C-K. Duck.
Duck.
It's named duck because head of isopod is like a duck.
Look like duck. They don't look at the colorful shape or the size of them.
Oh yes. Some species of isopod in other ocean is very big. And isopod can eat too. Some species is colorful and expensive.
If it's colorful, it's more expensive, right?
Yes.
Okay.
Okay. Now we know why you're interesting about the isopod, but how to set the optimum condition for pet it?
Again, please.
If I bought some isopod from you, how to set the condition that's optimum to pet it?
Just control humidity.
Humidity?
Yes, and temperature. You can breed isopod in box.
In box. Put it in box. Okay.
Yes, just put a soil, a dirt, a moss, and bark into it, and control humidity, temperature.
How can we control the humidity? Drain water?
Yes, just spray a water. Yes.
Water. And how to feed it? Feed the food.
We have foods of isopod, but isopod can eat leaf, soils. Yes.
Thank you so much.
Thank you. Thank you. Thank you.
May I invite Dr. Shontisha Jinaporn to give the certificate and take photos with the presenter in front of the backdrop, please. And the next presenter are from National Institute of Technology, Khosen Hakaratek College, with the project entitled Project Mamelo, exploring yeast from local flowers and the contribution to the local bakery. Please welcome.
Hello everyone, this is Yui and Kurumi from National Institute of Technology, Faculty College. Our research is Project Marmelo, exploring yeast from local flour and the contribution to the local bakery. Advisor by Professor Junya Kobayashi, Assistant Professor Osamu Terakado, and Ms. Ryoka Sakai. The purpose of our research is produce bread using yeast collected from the flour of Marmelo, and to revitalize the local community along with it. First, let me explain what a marmelo is. It is a yellow fruit growing Hokuto City next to函館 City, where our college is located, and has a pear-like aroma. However, it is a strong astringent. It has a strong astringent taste and is not suitable for eating. The challenge is that people of Hokuto City are trying to make processed marmelo, produce a specialty of the region, but the challenge is stagnant. We decided to add value to the marmelo and market it as a specialty of Hokuto City. There we have realized that we should focus not only on the fruit, but also on the flowers. Flowers have a variety of image and as a perspectives to the product. For example, some flowers have image of summer and energy. Therefore, we decided to develop product of the flower of marmelo rather than the fruit. The impetus for this research come from the experience of our college in producing circuits yeast collector for flowers, and with the completion of Dr. Kobayashi, his research was able to be conducted. Dr. Kobayashi, this research was able to be conducted.
The following procedure was
used to isolate from flowers. First, introduce the flowers into YPD medium and incubate at 13 degrees Celsius. Second, isolate the yeast on YPD agar medium. Third, isolate yeast on YPD agar medium. Here is the freeze-dried yeast. The result, yeast was fermented by the original method and tested for suitability for bread production. Sorry. The result confirmed for only one type of yeast was obtained for the forward obtained. The finished yeast was fermented by the original method and tested for suitability for bread production. Based on the rate of growth of the yeast and the condition with strong fermentation power. Feeling limitation to make only 1 hour one bread, we decided to provide fresh yeast to local bakeries to make bread for us. With the help of the city of Hokuto, we called on local bakeries in the city. Two Hershey's bakeries took on the task of making prototypes. We received feedback from each bakery and went through a trial and error process. And we could finally commercialize our bread in the city. With fresh yeast, the acidity was strong, but with dried yeast, the fermentation power remained the same and the acidity decreased. From this activity, we were awarded in the project-based learning contest. Finally, we hope that more people will become interested in Maumero through these activities. We also find that live yeast is difficult to handle because it is difficult to culture. Currently, we are continuing our research to mass produce dried yeast and to make better tasting bread. Thank you for your kind attention.
Thank you very much. You both are the effective presenter and also potential researcher. Okay, I like the project you work with. I have a question about the name of yeast, scientific name of the yeast you isolate from the flour. You know, the common yeast that we conventionally use for a long time is a Saccharomyces cerevisiae. And for your project, I did not see the name of them. Can you ask the name of them? Can you identify them?
Oh, we have no yet identification the type.
Okay, you haven't done the identification. Okay.
Therefore, we would like to make this issue
for the future.
Okay. Mhm. And how can you know they are yeast? The characteristic of yeast. Please explain the characteristic of yeast. Characteristics. Or the common morphology, common. Okay.
In your experience, in your experiment, it seems like you use YPD medium. Can we take a look? Can we take a look at the slide? Okay. And please tell us why do you prefer to use YPD medium? Yes.
Sorry, please again. Please again.
Why do you prefer YPD agar medium in this process? You use YPD medium to isolate, try to isolate yeast, right?
You use YPD medium. So the reason why do you use this medium? Okay. Because there are many media, many media that we can use to grow microorganisms, but you use YPD. So we want to know the reason why you choose this one.
Because it is the best way to culture yeast.
Okay, can I ask more about the marmelo?
Marmel.
Okay, because I am Thai, so I never try it. Okay, I just want to know is sweet or not? The marmelo is sweet or not? I never tried it before.
Sweet?
It's sweet.
Flavor. Yeah, sweet.
It's sweet.
Sweet flavor.
So it seems like that is sweet. It seems like it have many sugar in that fruit. Okay, so this is my suggestion. If it's sweet, but I don't know it's sweet or not. If it's sweet, normally it will have many yeast in their pure. So maybe it's another side to make research to it. Like you isolate it from flour. In flour also have a sugar in there, right? And their pure is also have many yeast too. Maybe it's make your study. You can isolate from there. Okay. If it's sweet, thank you so much.
The next, may I invite Dr. Buddhipong Wahakam to give the certificate to the presenter and take a photo. And the next project is color change and antibacterial mass filter from butterfly pea by using electro-spinning technique. From Princess Chulaporn Science High School, Phetchaburi.
Welcome committee and listener. My name is Khadisor Sangkhon.
My name is Wanpui Pianpan.
Today we will present about color change and antibacterial mass filter from butterfly pea by using electro-spinning technique. Currently, there are many diseases and physical disorders that are easily contagious simply by obtaining secretion into the body. For example, influenza or COVID-19 has affected people in the country due to the increasing number of infected people. As a result, people are turning their attention to masks that can reduce the risk when going out in crowded places. As we commonly see, masks are coated with anticipate coating from doubling of secretion. However, due to the COVID-19 situation, people need to wear the mask for a long time until accumulated of germ increasing that chance that the pathogen can enter the body. After studying the solution to the problem, we found extract called anthocyanin. It can change color when we evaluate change and anti-pathogen property. The project group uses above problem to make that mask filter that increase the discoloration property when exposed to secretion and anti-pathogen to remind user of the amount of secretion and also to tell user to change the mask by using anthocyanin fiber to make the mask filter. The purpose of our project is to make the mask filter that can change color and release germ, and most importantly, this filter can be used in daily life.
Material and method. First, we will extraction of substance of solvent from butterfly pea flower by addition. First, we will kill butterfly pea flower and soak it in the 50% ethanol for 8 hours. Then we will filter it within white cloth and filter paper to remove the residue. Then second, we will distill the ethanol with evaporator. We will use evaporator set speed at 100 and pressure 175 bar and temperature 60 Celsius. And we will put the substance extract 300 milliliters. Repeat it 3 times or until ethanol is evaporated. Then we will preparation PVA 60% weight by volume solution. We will mix 40 ml of distilled water with 6.4 of PVA, and we will prepare 5% of butterfly pea extract mixed with 60% weight by volume PVA solution. And the last is electro-spinning process. We will set electro-spinning process. The distance between the side link and the bender 15 cm and set speed 529 liters per second, and it takes about 1 hour. In the test, we have the 3 tests. We have dust in the air test, airflow test, and color value test.
The result of the dust measurement experiment we are divided in 3 categories: face mask, normal face mask, cold face mask, and anthocyanin fiber. In this image, little dust measurement in normal air has with a value between 150 to 200 ppm. And the graph on the left is a graph of normal phase mass, and the graph on the right is a that measurement of cold phase mass. Both graphs have similar ppm. This graph is a graph of anthocyanin fiber with a dot value is no more than 10 ppm. Second experiment is an experiment on airflow. We use airflow equation equal VA, and the box that suck air from the left-hand side of the image and blow it out of PVC pipe to the right side to measure wind speed. The normal wind speed is 3 meters per second. Normal phase mass and cold phase mass are insurmountable, but anthocyanin fiber has wind speed is 0.2 meters per second. When repeated in the equation, the table value is high, or the airflow is better than normal phase mass and cold phase mass. And the last experiment is a color value. L represent brightness, A represent green to red, minus A is green, A is red. B represent blue to yellow, minus B is blue, B is yellow. In the test, if its color difference is more than 1 and it fails, the color will change.
The question, all three types of matte filter have a dark position, but filter from modified peel fiber have the most because it's produced by electric spinning process that make the gap between the filter is smaller than other mass. And it has average LPG value 1 ppm, the average carbon dioxide value 7 ppm, and average smoke 5 ppm. And it has the highest airflow value, so it can beat easily. And it can change color clearly when touching silicon. In conclusion, the matte filter from butterfly peel attack have a low ppm and high airflow, made it easy to beat, and have an antibacterial and dark position. Thank you for your attention.
Thank you very much for your presentation. You have done a great job. Okay, your research is very interesting because it can be useful for medical application. So, but I'm thinking about antibacterial result. I didn't see the antibacterial activity result from your presentation. Did you work with?
In butterfly peel fiber have flavonoid, and flavonoid can antibacterial.
Oh, but actually, this is from the literature, from the reference. But
yes.
I mean, have you done the experiment on antibacterial activity of the developed mass?
We use the reference to improve that the butterfly peel fiber can antibacterial.
But it
no.
might be, how can I say, sometime when we have a look, the reference and read literature, okay, they may say butterfly pea plant can inhibit some bacterial strain, but in the real life, it might not work with the bacterial that flow in the air. Did you get my point? And we need to confirm. Sometimes the concentration or dose of the butterfly pea might affect differently. When you use only low dose, low concentration, it might not inhibit at all. not inhibit bacteria at all. But when you use high dose, okay, it might affect bacterial cell and make it damaged. But from your work, we don't see any concentration because you might choose only some suitable concentration for producing the mass, but that dose might not affect bacterial strain at all. So it would be better if you can test antibacterial activity from the fiber you develop for the next plan, next activity, next research. Yeah, you might try to use nanofiber you develop and check the antibacterial activity from them.
Yes, thank you for your help.
Okay, I will give you some idea. You can, that's the point that Ajahn Wuttipong already suggests you. You can use your mass that already developed, and you can test it like a filter, air filter. Okay, first you can first you will sterilize it in the oven or in the autoclave. In your school already have it, okay?
Yeah.
So when it's sterilized, after that you use it like a filter to test airflow, and you evaluate it after you as a filter, bacteria can pass through or not. You will can answer the question of Dr. Wuttipong. Okay.
Yes, thank you. Thank you very much.
Thank you so much.
Thank you, commentators and the presenters. May I invite Dr. Shontisha Jinaporn to give the certificate and take photos with the presenter, please? And the next presenter are from Nong Bua Pitayakhan School in the project entitled The Efficacy Study of Aerola calambola crude extract on bacteria.
Good afternoon, ladies and gentlemen. We come from Nong Bua Pitayakhan School, and we would like to present about the efficacy study of Aerola calambola crude extract on bacteria, and presented by Mr. Chanopap Nopawat, Mr. Thakor. Advisor is Mr. Tesathit Taruwan, Ms. Vijita Sripatyot, and special advisor is Mr. Kuntara Siriodom, and our team is Patdon Chai, not here. Yeah. Introduction. In the community of researchers, there are many of Aerola calambola, and the local flowering plant is rapid, and many fruits are waste. So our team is interested in using Aerola calambola fruit to make an extract for inhibit bacteria. Yeah. Aerola calambola is rich in vitamin C, which alleviates bleeding gum and blood urea disease. And it has high amount of fruit acid, and it can heal a wound. Staphylococcus aureus is a bacterium that inhabits the skin and mucous membrane of human and animal, and it can cause a viral infection such as boils, impetigo, postoperative wound infection, and the symptoms from this bacteria is fever, body ache, blistering rashes around the mouth and nose, and red skin eruption, and that can spread all over the body. Bacillus cereus, the symptom from this bacteria is emetic syndrome and diarrhea, abdominal cramp, and this bacteria can cause food contaminate. Yeah. General bacteria, some types of bacteria can cause the disease in human body, and our team is swab bacteria from leaf, hand, and doorknob. Variable, independent variable is crude extract from Arugula carambola. Dependent variable is inhibition zone. Controlled variable is positive control is tetracycline and ciprofloxacin. Negative control is dimethyl sulfoxide or DMSO. Objective to study the efficacy from to study the efficacy of crude extract from Arugula carambola to inhibit Staphylococcus aureus, Bacillus cereus, and general bacteria.
For experimental method, step 2, prepare the crude extract. First, we use raw foods of star food and slice, and then we dry it, and then we soak with 95% ethanol solvent, and next we filtrate, and then we aerate for isolate the extract and ethanol. And next we get the extract and we dry it again until thin and sticky, and then we bake it. Step 2, test our extract to inhibit bacteria by using agave diffusion method. First, mix crude extract with MSO, and then we prepare the nutrient agar. And next we spread bacteria all over the plate. And next we do the nutrient agar for 4 holes. And next we remove some nutrient agar that we do. And next we drop the substance for using micropipette for 30 microliters. And then we take it incubate and wait for the result. The results show that our extract cannot inhibit Staphylococcus cereus and Bacillus cereus, but our extract can inhibit general bacteria from leaf. Yeah. Discussion and conclusion. The extract cannot inhibit Staphylococcus aureus and Bacillus cereus because they are strong pathogens and they both have enterotoxin, and they are gram-positive. And Staphylococcus aureus are antimicrobial resistant. Crude extract. กรกมลปัตเต she used crude extract from live Arugula carambola with ethanol were effective inhibit Staphylococcus aureus, but our research, our team used raw food and make in dyeing process, which difference from above research. And next, but our extract can inhibit general bacteria. We didn't identify the general bacteria, and it may be gram-negative, like Vibrio cholerae and Escherichia coli, because Usa Ruamsuk, she used carambola food to inhibit Vibrio cholerae and gram-negative.
Next, suggestion, we should stain bacteria and find another type and concentration of solvent, and we should find a minimum inhibitory concentration and minimum bactericidal concentration. And this is our references. Thank you for your attention.
Thank you very much for your presentation. Okay, I have no doubt you have a strong presentation and communication skills. You speak very well. But some point that I want to give you for next presentation in the future. When you wrote down scientific name onto the slide, you must carefully check, is it italic? Because I see you did not make it italic, you know? Okay, so this is very important because scientific name must be italic when comparing with other font. Okay, so for the question related with your research, you conclude you cannot find the MIC for the Staphylococcus aureus when work with the carambo fruit extract, right? You use leaf extract or fruit extract something?
Okay.
Fruit extract.
From fruit.
Fruit,
Yes.
okay.
Yeah, from fruit.
So can you go back to please go back to the result slide. Okay. Here, okay. The maximum concentration you used here is 50 milligram per
50
liter.
milligram per milliliter.
This is the maximum.
Yes.
Why don't you increase up to 100 milligrams?
Because we use 50 milligram per milliliter because if it can inhibit, we not.
If we increase our concentration and 50 per milliliter is already can inhibit bacteria, and if we increase it, it's gonna be waste our extract to use for a product. Yeah.
So the data you show here is the clear zone, right?
Yeah.
Okay, clear zone from L1, L2, L3, L4. This is the common bacteria or general bacteria.
Yeah.
You isolate from leaf.
Yes.
Okay, so for table 1, please. No, table 1.
Table 1. Oh, sorry.
So you did not see any effect on Staphylococcus aureus and Bacillus cereus?
Yes.
Despite the same concentration. But you can't conclude that because these two species are pathogen. Because your fruit extract might affect them when using higher concentration.
Oh, we use a higher concentration, but
Is it?
have not
100?
100, yeah, but
Still not working with them.
yes.
Oh.
Because maybe we use raw food and we use dyeing process. Maybe some substance that can inhibit, maybe volatile. Yeah. We didn't gram. Oh, sorry.
Both of them are Staphylococcus aureus and Bacillus cereus is
Gram positive.
Right?
Yes.
And did you test the gram staining with the general bacteria? That is gram positive or gram negative?
Oh, we didn't. Yeah, yeah.
Maybe you can find or you can get the result why the table 1 and table 2 is give a different result.
Oh, yes.
Because gram negative bacteria and gram positive bacteria might have different response
Yes.
to the extract. Mhm.
Because of your conclusion, you discussed about the negative, right? The gram negative might be, you said your extraction might be effective to gram negative, right? In the conclusion.
Yeah.
So that if you make a gram staining with the general bacteria, maybe give your result.
Yes, it's in my suggestion too. Thank you so much. Thank you for your attention.
Thank you for the presenter and the commentators. Next, may I invite Dr. Vuttipong Mahakam to give the certificate and take photos with the presenter. The next presentation will start at 2:55 p.m. Waiting for 4 minutes. And if you want to go to the toilets are on the right side of the first and the second floor. And drinking water and snacks are also served in front of the classroom number 2301 at the right side. Thank you. The next presenters are from Sapporo Kasei Secondary School with the project titled Stink Bug Mimetics. Please give them a big hand.
So let's start our presentation. I'm Aiho and she is Aya from Sapporo Kaisei Secondary School. Our study title is Stink Bugs Mimetics. It's an original word, a cross between stink bugs and biomimetics. This is an example of biomimetics. Scientists made Velcro from cucumber hook. This means applying biological function and structure to practical use. So our goal is to utilize the stink bugs to make a rescue robot. In a disaster, the chance of falling down or being knocked over is high. Therefore, we want to make robots that can stand up by themselves. Our study purpose is find out how to stink bugs get up.
This is our previous research one, a kind of stink bug, Halimorpha haris, and sequential photos of stink bugs getting up. As you can see, Halimorpha haris hooks onto the ground with its feet, and it has a flapping motion to propel itself forward. This is our previous research too. Beetles have 3 ways of getting up, depending on type of the surface. First is using their claws, as well as short spine in the tibia and tarsus. Second is swinging their legs and wide body from side to side. Third is using their claws to grab onto another object.
Our hypothesis is that the hind leg stink bugs have a special structure. We think this because stink bugs use their tarsus when they get up, but other insects don't use their tarsus when they get up. We did 3 experiments to formulate this hypothesis. This is our experiment 1, observing by electron microscope. Our objects are 3 types of stink bugs and 2 other insects for compare from the stink bugs. These are results of observing of stink bugs. As you can see, stink bugs has 2 crowns and 2 spatulas on their tip of hind leg. We couldn't observe the revealed descent of this. We couldn't observe this because it's working from the tip of the tibia. And these are results of giant water bug and scurvy beetle. Sorry, we have a mistake on slide, so this sentence. They have only crowns, not half spatulas. They have one thing is common that they have cilia on their legs, so this. Stink bugs and scurvy beetle have a structure as longitudinal striation inside their crowns, but we didn't see it on the giant water bug. This is discuss of experiment 1. We found that stink bugs has a special structure that is crowns and spatulas. So we consider that they use them. To get up.
Experiment 2 observed how stink bugs getting up. We prepared a 1 mm thick colored sand on petri dish. Then we turned the stink bug onto its back and we observed it. In addition, we observed the patterns it made in the sand. However, the stink bug didn't get up. It just dig in the sand with its hind leg. So we changed the thick and fineness of colored sand. We prepared regular colored sand and ground colored sand. And we put on these colored sand on petri dishes, 5 mm thick each. Then we observed again. Please look at the upper image. Leg markers are parallel to each other. Please look at lower image. Sand powder put on their hind legs, especially tarsus. Discussion of experiment 2. The stink bug uses its unique spatula structure to push itself up on the ground. In order to get up, the stink bug needs its rough surface, and which can be useful for practical use
in robots because disaster-stricken areas has many uneven surfaces. Then this is our experiment 3. We have 5 objects and 5 entries, 3 types of stink bugs and giant water bug and scarabaeater. This is a result table and average data of stink bugs length. We set the ratio of horizontal length as 1, and sometimes we couldn't measure tails because it was broken. These are all insect results. Stink bug and scarabaeaters can get stink bugs and scarabaeater can get up themselves, but giant water bugs cannot get up themselves. We found the ratio of vertical length and horizontal length of stink bugs and scarabaeater is 1 to 1.9, while giant water bug is different at 1 to 2.7. We found the common ratio of the stink bug and scarabaeater is 1 to 1.9. We think this ratio is probably the most efficient ratio for getting up. So our future outlook is to create a rescue robot that can get up on its own. And we would like to continue our research for practical application. Thank you for listening.
Thank you very much for your nice presentation. Your work is fantastic and awesome. I enjoy listening to everything that you have to say. Okay, great job. Yeah.
Thank you.
So I have only one point I want to know from your experiment. You mentioned about spatula structure that can help to getting up. Okay, spatula structure. Okay.
Yes.
These 3 species have different spatula structure, right?
Yes.
So which one is better for help them to climb up? From this result.
But we didn't research it, but spatula structure can be help to getting up.
To get up.
Yes.
So which one is better from your opinion?
I think
because you said in the future you want to develop the robot model from this structure, right?
Yes.
This is a biomimetic. So when you have a look all result you done, how can you create the robot from this data?
We think.
Imagine you going to create.
We think
A
at
robot.
first we want to make a model of this spatula structure as a as a 3D model in digital. Then we create a we create a plastic model by computer. Then we will talk to the advisor of robot. Then we want to make a but practical application. Yes, we think so. Yes.
From your result, you observed this bug and you said it cannot get up from the sand, right?
Yes. 1 millimeter thick.
Okay, okay, it cannot get up from the sand. But in the future, you will create a robot. Okay,
Yes.
how you develop it? If you want it to get up from the sand too, how to develop it?
We want to, at first, we want to make model by myself. Then we will text to the professor of robot. Then we communicate to the professor. Then we will make a robot.
You need to get some idea from your professor, right? Okay. Okay. Thank you very much for your nice presentation.
Thank you.
Next, may I invite Dr. Shontisha to give the certificate and take photo with the presenter, please. The last project is Effects of Weather and Soil Quality on Bamboo Growth in Tokyo from Tokai University Takanawadi Senior High School. Please welcome.
Hey, hello. I'm from Tokai University, Takanadai Senior High School in Japan. I'm Masahiro Tsunemi, and my partner, Kokoa Oku, is today. I will be presenting about effects of weather and soil quality on bamboo growth. For today's topic, we'll be talking about the objective, methodology, results, and our future study plan. We're entering introduction, bamboo grows well, better than trees, and overall. And bamboo is used in many ways, such as food categories, furniture, baskets, charcoal, and bamboo pulp. For our experiment, we have used Busa multiplex var agalagams. For our objective, we want to study the effects of weather and soil qualities on bamboo growth to find out the best way to grow bamboo. For our research question, we have the following. Does weather conditions affect bamboo growth rate? And if soil qualities or radiation affects bamboo growth rate? For our study site, we have studied at Tokyo, Japan, Kanagawa Prefecture, and Sagami Lake. For our material equipment, here's are the following. Flexible tape, Globe Observer app. This app is used to measure the length of the bamboo by using trigonometric functions. Thermometer, tray, frying pan, scale, beakers, distilled water, oven, NPK, which stands for nitrogen, phosphorus, and kalium, and pH test. And for the second page, we have a lux meter to measure solar radiation. And for our methodology, we have the following to study the weather conditions, soil quality, sunlight intensity, and the bamboo growth rate. For our weather conditions, we gathered air temperature, humidity, and rainfall. And weather data will be collected daily during the experiment from June to September 2023. And for bamboo growth, we measured the circumference, the height, and the surface temperature of the bamboo. And also, we studied our sunlight intensity. For soil quality, we measured the following: the organic matter, moisture, the surface temperature, inside of the temperature, and soil pH elements. From this on, these are the results. And the first slide for weather conditions, the left graph is for air temperature and humidity, which shows from June to September. And for the right graph, we have rainfall at the study site. And for the next slide, for bamboo growth, this is the bamboo height. And on the right side, we see the circumference of the bamboo. Here are the soil qualities and each elements found at each site. These are the table. And when we briefly look at it, we find that Site 2 has the highest temperature record and thus the highest growth rate from June to September, which made me think that in Thailand, bamboo grows well in Japan because of the high temperature in Thailand. For Site 5, we found the average soil quality found for each segment. For Site 6, we found the highest amount of nitrogen and it was the most acidic. And thus, Site 6 had the lowest growth rate, which led to believe that high amount of nitrogen and low pH may be bad for bamboo growth. And for our examination, we found the growth condition for bamboo growth. Higher temperature can lead to better growth. And for precipitation, we have less data, so it's to be determined. And for the following, we'll connect to the next slide of our realization. As I said before, lower nitrogen can lead to lower growth rate, and warmth and not too much sunlight, which we see in the bottom. We see that for Site 5 and 6 is 81 watts per meter and 73 watts per meter squared, which has lower growth rate than the lowest radiation that is found in Site 2. And excessive sunlight affects growth, and that is connected to number 2. And we also found that if we split the data evenly, we found that rainy parts tend to affect growth the most, which means that when it rains a lot, bamboo grows a lot. And this connects to why bamboo grows a lot, which made me think that Thailand is rainy and led to believe that Thailand has bamboo that grows more than in Japan. For this research, we also like to thank Princess Chulalongkorn Science High School Trang, as we did a collaborative research last year. These are one of our first graphs we made, actually. When our first experiment, we did not know the exact number of each elements, trace, low, medium, and high, but our last one, we know exactly the exact amount of each element found in each site. And for our graph, on one of our first experiments, we did not have enough length to experiment, but on our last one, we were able to do a contested research for 3 months straight. So we would like to thank the collaborative research that has led us to step up for the next experiment for our future study plan, we would like to increase the number of sites. As you might have realized, there was only 3 sites of bamboo, which means we only measure 3 bamboos. So we would like to increase the number of sites in order to gather more data to find a concrete conclusion. And for second, we would like to increase the length of the experiment so that we can measure the bamboo for extensive research and also for more data. And for the third, we would like to find a more exact way to find organic matter, because when you look at the organic matter on the first top 3, we see the same numbers for phosphorus and kalium for 3 straight sites. And addition to sunlight, we would like to explore the aspects of water, because we believe that water quality affects the most. Here are our references we use for our experiment. Thank you for listening.
Thank you very much for your nice presentation. Okay, I like the way you deliver your presentation. You have strong communication skill. Your slide is clear, concise, and beautiful. Okay, I love it. Okay.
Thank you very much.
Okay. All right, so for the question related with your research, I curious to know what is the best factor, the best factor, the best environmental factor
The most
that
important.
that can help improve bamboo growth from your experimental data.
Okay.
Suppose you will give the knowledge from your research to farmer. They want to grow bamboo and get higher growth, higher yield. So can you give some data to them to improve the bamboo growth from your soil quality data?
So I think the
how can
question
they
is
improve?
like, what's the best way to grow bamboo? For conclusion, so when you look at this chart, like I said before, higher temperature leads to better growth. So I believe higher temperature is really needed to help growth bamboo. And also, we also found it very interesting that higher elements of nitrogen can lead to lower growth. So when you fertilize the soil, we would suggest to not put too much nitrogen in, or else it will not grow as much. And also, acidic soil can lead to lower growth rate. So I believe that I can't say like the most important thing. I think it's a lot of all of the components that combine together that helps bamboo growth. So all of them I think are important for and necessary for successive bamboo growth.
Okay, so I got your point, but some factor difficult to be controlled. For example, nitrogen and phosphorus. Okay, especially nitrogen, because it can lower the growth of bamboo, right? But when we grow bamboo until the mature stage, they have a lot of leaf, and then they fall onto soil, and the soil get nitrogen source from leaf again. So how can we control this?
Yes, so when you look at the bamboo growth, I've only done up to 10 meters from the 3 sites I've done. So I'm jumping to conclusions, but for example, let's say we're starting to grow bamboo from a shoot. So I would suggest that controlling nitrogen when you're controlling it from shoot would be the best possible way to grow bamboo. So after it has grown to a certain amount of height, we might suggest to put it in a different soil so that it can grow leaves, good leaves, and a strong trunk.
Oh, thank you. That's all I want to hear from you. Thank you, Rod.
Thank you.
It seems like you're very interesting in bamboo. And the last year you already have a research in Thailand?
Yes. Actually, 1 year ago, we did a collaborative research. They're actually sitting right there with us. Yes. Yeah,
my God.
we did them together.
Okay. And and now you study in this research is study in Tokyo.
Yes, studied in
Okay,
Tokyo.
and it seems like if I'm not mid your lecture, it seems like you have a 6 side to study. 6 or?
Yes, well, we started with 6. And
Can I take a look at map? Okay. I just want to know, in each side is have an environment or the area is same or different?
Actually, I haven't shown the exact place for each bamboo, so thanks for the question. Each bamboo has different places, so like I said, it looks like at the same from this perspective, but you look at each side, they're at different, it's like a stair, and different
high from a sea, high level from a sea.
Yes.
Okay. So which one is you prefer to grow a bamboo?
Once more, please.
If you are owner of this area, which side you prefer to choose to grow a bamboo?
Okay. So back to the slide. Site 2 has the best growth rate and the best circumference. So I believe that Site 2 is the most biggest bamboo in general. So I believe Site 2 is the best bamboo. Thank you.
And may I invite Dr. Wuttipong Mahakam to give the certificate to the presenter and take photos. And after that, we would like to invite everyone to take a photo in front of the backdrop. I would like to invite everyone to take a photo together.